have you come up with questions in the meantime? Then I would like to start a bit of an excursion. This is not directly classification, because you cannot only do classification in life. And we will return to classification next week. But for now, let's do this short detour, which I take as an excuse to introduce you to some very recent literature that I find interesting. This is still about describing dependencies. And this time, describing dependencies with the help of Gaussian graphical models. So graphical model is a shortcut for probabilistic graphical model. And probabilistic graphical models are sometimes summarized as the espousal of probability theory and graph theory. This is effectively becoming a very convenient language for modeling in data analysis, in machine learning, and this is currently a very active field of research. And in Heidelberg, there's a research training group with more than a dozen PhD students who are now focusing exclusively on graphical models. The reason they have become so popular is that they are a very succinct means of summarizing a model. So I will draw a couple towards the end of the semester, and you always draw small bubbles and friendly lines, and the expert from those lines, or rather the missing lines, can read out what class of model you are talking about. And in particular, missing lines means or show you where you have conditional independence, and these conditional independencies can either be estimated from raw data, or they can be injected as prior knowledge, and this dramatically reduces the number of parameters that you then need to estimate. So throughout this short hour, I will assume that the observations that you have are multivariate normal, and a prime example of where you can use this is to find pathways in systems biology. But this extends way beyond biology. You could also say that perhaps you have taken over a chemical plant from somebody who has ill-documented the whole construction, and you can now tweak on different, or you can, well, tweak in different places and get measurements out, and you want to find out what is actually connected to what. This is a similar kind of problem. So in this example, finding pathways in systems biology, you could assume that we have n individuals or n conditions, and p gene expression levels. And let's further assume that p is much larger than n. So to give you an idea, in typical research, p can be of the order 10 to the 4, 10 to the 5, 10 to the 6. N will be 10 to the 2. So with other words, you may have, depending on what exactly you have measured, but you may have 100 points in 10,000 dimensions. And with 100 points in 10,000 dimensions, it's not easy to do anything. And this is why people make assumptions. One of them being that, as I said before, in this entire section, I will assume that the data has a multivariate normal distribution. So people will assume that your observations are samples from a Gaussian. By the way, that I've chosen this particular example is not accidental. because the appearance of microarray chips, these chips that allow you to relatively easily measure gene expression levels for hundreds of genes at relatively low cost. This was a huge boost to biology, but especially a huge boost to this field of statistics and machine learning, so to deal with few observations in high-dimensional spaces. And the first thing that people did with that kind of data is to try and identify correlation graphs or relevance networks. In correlation graphs, you try to visualize only the marginal independence structure. So you simply compute the Pearson correlation coefficient between each of your random variable and any other. And if the correlation between two random variables is below a threshold, you will say that, okay, this is roughly 0, and you can then assume marginal independence. So what you do here effectively is to find zeros in the empirical covariance matrix. A possible finding could be that you identify a covariance matrix which has a block diagonal structure, and then you would say, okay, there are two non-interacting systems in my data. But in general, the structure of the covariance matrix or the locations where you do and don't find zeros can be much more complicated than just that. Now, more interesting, there has been an alternative proposal, namely to construct concentration graphs rather than correlation graphs. They have been reinvented many times because they're so important, so they're also called covariance selection networks. Or Gaussian graphical models. Rather than finding zeros in the correlations as before, you now try to find zeros in the partial correlations. And I'm explaining what those are in a minute. These partial correlations have long been studied by people in the time series business. So there is a class called Autoregressive Models, and you could, for instance, now don't copy this down, you could, for instance, construct a time series which depends somehow on the previous, so alpha times plus some kind of random input. And if you specify this kind of model, then you will, so x is a random variable, then you will obviously have some correlation between y at instance t and y at instance t minus 1. But you will also have, because t minus 1 is correlated with t minus 2, you will also have correlation between t and t minus 2. But this is somehow a correlation which comes only through this parameter. And the difference between these two kinds of correlations that I mentioned is the difference between correlations and partial correlations. So a partial correlation between two random variables x and y given z is the correlation that you find in the residuals after regressing both x and y on z. So x and y are single random variables, but z can be a collection or a set of random variables. And if we're interested in the partial correlation, we will first set up a linear regression trying to explain x in terms of the set of random variables z, and then trying to explain y in terms of z, and then we keep only the residuals and compute the correlation between these residuals. And we then find a partial correlation coefficient, which can be written as rho between x and y given z. And this partial correlation coefficient is related to the inverse covariance matrix if the data has a Gaussian distribution. So just like the normal correlation, this is a covariance standardized by well, the square root of the product of the variances. And that is a result which takes a page or two of algebra to prove, which I'm not doing now. Corresponds to element xy of the concentration matrix. Similarly normalized. With K being the concentration matrix, which is a new technical term, or so it is not concentration divided by, but it's an underlined. It's a concentration matrix, or precision matrix is a different word for that. And that is just the inverse of your covariance matrix. So if you assume that your data is multivariate Gaussian, so if x and y and z, which can be a set of random variables, if they're jointly normally distributed, some mean and some covariance matrix, then x is conditionally independent of y given all the variables in Z, if the element xy of your precision matrix is 0. So before we were trying to find zeros in the empirical covariance matrix, and now we're trying to find zeros in the empirical inverted covariance matrix. So I'm going to show an example. Let's say we have 16 random variables. These are not quite 16, but so let's say these are 16. And I have here drawn an edge only between those random variables that have a non-zero partial correlation. Then I could, for instance, say that the first and the second one are correlated, and sorry, there's a minus missing here, which is important. I could say that the first and the second one have a non-zero and positive partial correlation. The second and third one have a partial positive correlation, and so on. So if I'm drawing the precision matrix here, this will be a banded matrix where I have diagonal entries, and then I have one off-diagonal entry, namely for the left and right neighbor. And because the ring is closed, I have also an element appearing here and there. And if I have partial correlation between the first and the second and third, it also follows that they are correlated. So let's look at a numerical example. I've taken 16, not 17 as is written there, Gaussian random variables arranged in a circular graphical model as you've just seen. And I have plotted here the precision matrix. So it has exactly the banded structure that I have described. And if we now invert this to obtain the covariance matrix, you will see that well, actually, I have a few elements which are precisely 0 here, I think. But overall, through of matrix inversion, this band here smears out, and we see something all across the covariance matrix. So the interesting point, I think, is that when we, if you remember what a Gaussian looks like, So I'm omitting the mean for now. So we have x sigma to the minus 1 x, or using the concentration matrix, we have x transpose k x. And if I now explicitly introduce these interactions between neighboring random variables, I obtain a very compact expression for my concentration matrix, or I can formulate this as an energy function. However, the covariance matrix that I introduce or that I induce will not have the corresponding sparseness. And it is a property of the matrix inversion that associates this sparse precision matrix with this non-sparse covariance matrix. Now, the minus was important because if you look at the color bar, you don't see the numbers on this color bar. If you look at the okay, let's do it the proper way. So, if you look at the numbers on this color bar here, you will see that these off-diagonal elements that I introduced were positive. So I have positive elements here in my concentration matrix, and that corresponds to negative partial correlations. And if you now look at this covariance matrix, along the diagonal, I see the variance of each of these random variables that I plotted in a circle. And you now see this zigzag pattern here of negative values. So we have anti-correlation, and then correlation, anti-correlation, correlation, and so on. Okay? So if between these two variables I have anti-correlation, and between those two variables I have anti-correlation, so it means that this is, let's say, a random variable number 1, 2, and 3. Then if 1 is positive, typically 2 will be negative, and vice versa. But let's say if 1 is positive, then in all probability 2 will be negative. And because these are also anti-correlated, 3 should again be positive. So I should have positive correlation between 1 and 3. And this is indeed what you observe in this matrix here. I have a pattern here of, well, those are the variances, and then I have negative, positive, negative, positive, negative, positive correlations, okay, that propagate around this ring. Now this ring, all right, this is somewhat artifactual, you know, who would come up with this model? But in fact, similar models are used in image processing. And I've used this ring here just because it gives me this very nice primitive structure in my precision matrix. What people would do in image processing is that they would consider not a ring of random variables, but they would consider a grid of random variables. Okay, and because I work in image processing, you will have to bear with me sometimes introducing examples from this field. And now these Gaussian graphical models have two applications. I can either insert them as a prior, or I can try and estimate them from the data. In this image processing example, I introduced them as a prior. So, for example, let us say that I have a parameter estimation problem. I want to find the most likely set of parameters given my observations. And then by Bayes' theorem, I will say, okay, this is proportional to the probability of my observations given those parameters times a prior on the parameters. So the observations is what I have, the parameters is what I want to find, what I want to estimate from the data. So, you know, you can think of a physics experiment and you want to measure something on a field, or you can think of medicine where you want to identify the parameter, for example, from a time series of an experiment where you injected a contrast agent or so. So let's say we have some noisy data and we want to estimate those parameters. Then we need this term here. This is a likelihood. This will tell you how probable a given observation is for a particular parameter value. This is something you need to model. For example, well, you know, domain knowledge goes into here. So if you're in physics and if you want to measure the set of parameters, you should be able to, in a generative sense, define how probable it is to make a particular observation somewhere at your lattice given the parameters. And then on the other hand, you have a prior on your parameters itself. So the typical prior that people put in here is to assume that the parameters will be relatively smooth. And in this case, you can formulate a precision matrix just as I have done, except you will probably use negative entries on your off-diagonal so as to obtain positive correlation in your covariance matrix. Okay, and then there's much more to that, because typically this model is factorized such that you assume that the observation at a given point in your lattice only depends on the parameters value at that given point in the lattice and so on. But main point is you could construct a prior for your problem which in the spatial layout has interactions that are depicted here, and that will give you a corresponding precision matrix. And if I have a two-dimensional layout, I obtain some kind of, you know, more off-diagonals here. And to avoid all these complications, I've just used the ring in this example. However, the other use of this is coming back to that example here, where you don't plug in the structure to bring your prior knowledge to bear, but where, on the contrary, you try to estimate the underlying structure. So you just have a sample of observations, and you would like to know what is the underlying model. So to make this, you know, very graphically obvious, you can ask the following. I have observations for these different random variables, and I now want to know which ones are truly associated, or which ones have non-zero partial correlation. Okay, you're trying to understand the structure of your network. And I want to show you an example for how people do that. This is from a biology paper. So this is E. coli data, paper by Schäfer and Strimmer. And I don't even know what exactly this is. They had some kinds of measurements, gene expression levels or protein expression levels. And they tried to understand which of these things actually relate to each other. And to that end, they estimated these partial correlations. And dotted edges here are negative partial correlations. So things that will result in positive correlations, and vice versa. So when you have a positive partial correlation, this probably means that these components inhibit each other, that they are playing against each other. All right. Now, when you try to estimate a graph, as I have just shown, from observed data, you are confronted with two kinds of problems. first, if indeed your number of observations is smaller than your number of features, you will have singular covariance matrices. Because remember that what we can estimate from the data directly is just the covariance matrix, but we're looking for its inverse. And if the singular covariance, if the covariance matrix is singular, for example, when n is smaller than p, you run into difficulties. And secondly, you might, of course, have statistical or stochastic fluctuation in your data. So you might by chance find a non-zero partial correlation, even though the variables are not related. And to both ends, people use regularization. I've used that word before when we talked about stabilized versions of LDA just a short while ago, or LDA and QDA. Regularization means you somehow want to stabilize your estimation, typically by injecting prior knowledge that simplifies the estimation problem or makes it more stable. And in particular now, I want to mention stability selection. This is a paper from Mainzhausen and Bühlmann. And this has caused quite a stir already. And I think this will also turn out to be one of the really important papers of, well, it was published last decade, so I think 2009. So let's say one of the really important papers of the end of the last decade. It's a very simple recipe, namely you repeatedly sample from your data. So you're not using your whole data set at once and doing the estimation once and then there's nothing else to do. But instead, you repeatedly sample n half from your training set without putting back your samples into this bin. Okay, so out of your one training set or out of your one data set, you thus create many bootstrap data sets. And secondly, you use a regularized estimate of the precision matrix So the graphical lasso was presented by the Stanford people. So you use a graphical lasso which does an L1 regularization, and I will explain only later in the semester what that is. So use graphical lasso to estimate a sparse precision matrix. So we inject a little bit of prior knowledge that all things being equal favors a sparse solution. And we will really look at the algorithms behind this around the middle of this semester. And now you have generated your many bootstrap data sets. You have estimated a graph on each one of these. And now you do summary statistics and you check how often did a particular edge appear in one of your bootstrap data sets. So you only keep edges that appear in a significant fraction of your newly generated data sets. And I'm going to show the by now famous picture from the paper, which is this one. So we see from left, so this is again a data set from biology. Each tiny dot here represents one protein, I assume, or gene. And the question is to find out which genes are playing with each other. So, you know, if you don't do any data analysis, you can either assume that nobody talks to nobody, and you have no connections here whatsoever, or you can assume that everybody talks to everybody, and then you would have, you know, this full combinatorial set of connections. Now, you want to know what is the truth. And using a Gaussian graphical model, so you assume that all of your observations are jointly normal, you can use, for example, just this sparse estimation that I mentioned by means of the lasso. And you have a tweaking parameter lambda, and the larger lambda, the sparser your model becomes. And you see that for a reasonable range of lambdas, you find those models. So those suggestions for which variables have partial correlation. So where should you draw an edge or a connection in your graph? And down here is the result that you find when you use this graphical lasso procedure on each of the bootstrap data sets, and then just check for which edges are stable, which are recurrent, hence also the name stability selection. And what is very nice is that this procedure is less sensitive to the choice of this parameter lambda. So those sets of edges found are almost the same, irrespective of the precise value of lambda chosen. So stability selection itself seems to be an idea that will be applicable to other problems also. So rather than use your whole data at once, resample from your data, fit a model on each of these subsets, and then try and find what parts of your model are consistent across these subsets. This idea seems to carry over to many domains, but in this paper they've also been able to prove quite a few properties for this particular scenario here. For the scenario of finding between which random variables you have non-zero partial correlation. All right, so overall in this last hour, what we looked at were jointly normally distributed random variables, however, with some interesting structure that you can either impose a priori by saying, I believe that I have similar values between individual pixels on my lattice, or structure that you can try and infer from your data. As in this biological example, because in our body, you know, not every gene is talking to every other. That would be overly complex. But some genes must be talking to some others, or else we wouldn't exist. And this is a means of trying to find out here with very little effort. You know, you just get a large table of data, and you can in this way find out which players are actually, which players seem to be talking to each other. Any questions? So parts of this we will do in more detail later in the semester. In particular, I mentioned that the conditional of a jointly normal distribution, that's something that where we will really do all the math later in the semester. Also for the lasso itself, we will do all the math later in the semester. But the stability selection, I just wanted to introduce it to you without giving all the details. because I think this is an important method that you will likely encounter again if you have anything to do with this field. Okay, next week is Easter Monday, so we will have exercises as usual this Wednesday, and then 1 week later on Wednesday, well, we could do a lecture for 2 hours, which may not be such a bad idea because it's a short semester, because we have another public holiday on a Monday. Or we could just have a Q&A with Anna in this case, so where you can ask any questions on anything you've seen so far. Who wants an extra lecture? Okay, modest. And then, okay, I don't ask the others. So we will have a normal exercise this Wednesday, and then Q&A in the following week. See you then.